Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Jeffrey Meyer will present Neuroimaging Inflammation in Clinical Depression and Obsessive Compulsive Disorder. The Brain and Behavior Research Foundation funds the most innovative ideas in neuroscience and psychiatry to better understand the causes and develop new ways to treat brain and behavior disorders. These disorders include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, and schizophrenia. Since 1987, the Foundation has awarded more than $394 million to fund more than 5,700 grants to more than 4,700 scientists around the world. 100% of all donor contributions for research are invested in our grants to scientists. I'm delighted to introduce Dr. Jeffrey Meyer. Dr. Meyer is head of the Neurochemical Imaging Program in Mood and Anxiety Disorders at the Center for Addiction and Mental Health in Toronto, Canada. He was a 2015 Foundation Distinguished Investigator Grantee and a 1998 and 2000 Young Investigator Grantee. Today's webinar will begin with his presentation. This will be followed by a question and answer period. To submit your questions, please use the Questions tab on the control panel on your screen. Please feel free to submit your questions throughout the presentation. And now I'm pleased to introduce Dr. Meyer. Jeff, the floor is yours.
Well, thank you so much for that kind introduction, and thank you, Dr. Borenstein and the Brain and Behavior Research Foundation for inviting me to present today. It's really a pleasure. A number of findings have been really quite exciting in the last period of time because we have these new advances in neuroimaging markers of inflammation that really weren't available in the past. So this is really going to be useful for addressing key questions that can be important for clinical impact. Today what I'll do is I'll be presenting some new findings that have occurred just over the last few years and discuss their impact. Just before I get going, I'll just acknowledge conflicts of interest. So with markers of inflammation, typically when we make markers, we look also for blood markers of inflammation to relate to the changes that happen in the brain. And as a result, we tend to patent these as a way to try to make them more widely available to people because then companies can take them up and then distribute them. First, I'm going to talk about the clinical challenges of treatment for major depressive disorder and obsessive compulsive disorder. Then I'm going to move into talking about imaging inflammation with positron emission tomography, a type of imaging tool that can be quite highly sensitive for detecting inflammation. Then I'll move on to talk about a form of imaging called translocated protein imaging in clinical depression. And when I'm talking about clinical depression, I'll also refer to it as major depressive episodes or episodes of depression and major depressive disorder, reflecting repeated episodes of depression. I'll talk about imaging of inflammation and obsessive compulsive disorder, some interpretations and implications of using this imaging for improving clinical care, and some of the technologies and advances that are ahead. So a key issue with major depressive disorder is that it's very common, with 4% of the general population amidst of a depressive episode, and at least 50% of major depressive episodes don't respond adequately to treatment. With respect to obsessive compulsive disorder, a key problem is that it is reasonably common, with 1 to 2% of the population being affected by it, and as well, a third of people with obsessive compulsive disorder don't respond adequately to the best evidence-based pharmacotherapies, which are also antidepressants that raise serotonin. So a key question for these conditions is, do antidepressant treatments that get used for these conditions actually reach the target, and could that be an issue for having non-response? So what I've shown here on this slide is an imaging study, and what I've done is I measured a scan that looks at the binding to the serotonin transporter, and we scan before and after a 4-week treatment period. And what we do is we look at the percent of change in the binding measure that we get, which reflects blocking of the medication for the site. And while here I've shown the striatum as the region of occupancy, all the regions of the brain tend to have very similar occupancies. So that's what we've got here on the y-axis, the percent of sites blocked, and on the x-axis we have the dose that was administered. Now in this study, people who had depression were given minimum therapeutic doses that would typically treat depression, and then people with concurrent psychiatric illnesses like obsessive compulsive disorder would get higher than usual doses that would be appropriate for the condition. Because we didn't want to treat people who had clinical illnesses with subtherapeutic doses, healthy volunteers took subtherapeutic doses to get the lower occupancies that were found in the curves. And while there's a wide range of potential occupancies that can happen with these medications, what we found is that doses that distinguish from placebo that we typically had about an 80% occupancy. So if one gives the right dose of medication, there's not really much of an issue of reaching an adequate amount to the target. And if one raises the dose, you get more occupancy. So now you may look at the graphs and say, well, gee, paroxetine, a minimum dose, is the odd person who has a lower occupancy, or over here with citalopram, or over here with fluoxetine. However, if you look at the same data from the same study and you look at the plasma level in the blood, and you look at this in relation to the occupancy, what you can see is as long as there's enough medication in the blood, there's enough medication reaching the target in the brain. So the issue of having non-response is unlikely to be related to engagement of a medication reaching a target. And that has implications for conceptualizing why might it be that people don't respond to treatments. And what I'm showing here in this slide is a conceptual framework of how to look at the idea of non-response and an underlying concept. So what I've got here is an idea that psychiatric illnesses will affect circuits in the brain that we normally use for functions, and those circuits don't function the way that you would like them to, leading to the symptoms. In some illnesses, like in some areas of medicine, you have one disturbance or injury, and that's the main injury that causes all of the disease. But in the case of psychiatric illnesses, what we think is that there can be a number of different causes of neuronal injury or neurochemical disturbance, which I've labeled here as A, B, C, D, E, and F. Here is saying perhaps there could be 6 common sort of neural injuries and then disturbances, and that in someone who is low risk for getting a depression or obsessive compulsive disorder, they may have none of these potential neurochemical disturbances or injuries within the circuits that are implicated in the brain for these illnesses, whereas someone at high risk who can have for the illnesses might have one of these changes, whereas someone in the midst of an active episode could have a certain combination of changes within the circuitry that's affected by these illnesses, or they could have a different combination outlined over here, or an alternative combination. And then someone who happens to have a comorbid illness might have some changes that are in common with the other illness, but would also have some changes that would be different. And so the reason that we think that it could be non-response is that the conventional antidepressants might only reach a couple of these targets, sparing other targets, and therefore not leading to a response. It's a problem of mismatch, which makes it particularly important to be using imaging as a strategy to try to identify markers reflective of phenotypes or changes in disease, and then continue to use imaging as a tool to assess therapeutics that can engage these targets, develop low-cost surrogate predictors of what's found in the PET scans or whichever brain scans one happens to be using, as they're typically overly expensive to be using routinely for clinical care, and then to look at the effects of taking a therapeutic that engages a target, matching it to someone with illness who actually has that alteration in their illness that corresponds, and then seeing whether one can get a better cure and a higher likelihood of outcome being favorable. So I'm going to talk about positron emission tomography scanning, which is a very sensitive type of scanning, which is the focus in today's presentation. So with positron emission tomography, you have this camera with the ring of crystals, and people go into the scanner and they get a radio tracer, and the radio tracer or dye goes into the brain, and it lets go of a positron that collides with an electron. The electron's already there. And then you get these two emissions that are in coincident in opposite directions. And because you get these two emissions in opposite directions that get detected by the crystals, it allows for a high level of localization and is a very sensitive technique. So the sensitivity for positron emission tomography is extremely high. The challenges are to make tracers that can be suitable for positron emission tomography, and that can be a multi-year process. But when you have these tracers, they can be very useful for key questions. The experience for someone going in to a study is to lie on a bed, a mask is slipped over the face, they're slid into the scanner. Sometimes we take blood samples at the same time to aid in the quantitation of the tracer and where it's gone in the brain, and look at that relative to blood. Initially, the transmission scan that's done to correct for tissue that is present between, it's a correction factor that's applied. And then the radio tracer is given, and the scanning takes a while. It takes about 90 minutes to 2 hours because we need imaging of the tracer going in and out of the brain over that time period in order to develop the measures of binding that we obtain. So when I'm talking about inflammation, I'm talking about the bodily response to infection or trauma. So everyone's had a scrape on their skin and gets a hot red response, and our bodies can use this as an adaptive tool, but sometimes they can overdo it. In the brain, a key participant in inflammation are microglia. They're about 7 to 10% of the cells in the brain, and usually they are sitting around with a smaller cell body. They have these long dendrites, and they're in a sensing, detecting sort of mode, scouting away. But when they detect evidence of pathogens or of trauma, and they detect molecular patterns corresponding to these, they become activated, so the cell bodies become larger, the dendrites become shorter and thicker, or they can even be amoeboid, and they overexpress translocator protein when their morphology changes. And that's of particular interest because our scanning dyes and techniques bind to this translocator protein. The translocator protein is an 18 kilodalton protein that crosses the mitochondrial membrane 5 times. It can sit as a homooligomer in chains, or as a monomer on its own, or in a heterooligomer with other proteins, which is often how it's described. But our main interest in it is because it's overexpressed when microglia are activated. It has other roles. It translocates cholesterol from outer to inner cell membranes, and it also influences mitochondrial function. If, for example, you give a drug that binds to translocator protein, then you can affect sometimes respiration or predisposition to apoptosis in cells. Now, the translocator protein is also not completely selective. It also is found in other cell types. So it's found also in astroglia, another cell that can participate in inflammation, and endothelial cells. But during most inflammation challenges, it seems to typically represent activated microglia. So, for example, in rodent models that have been studied after giving toxins or inducing strokes or giving a sugar fat substance lipopolysaccharide that elicits an immune response, the changes in translocator protein binding are typically associated with the markers of microglial activation rather than changes in astroglial activation. So it can be possible of exceptions for this. For example, if someone happened to have an astroglial tumor, then yes, you would get more translocator protein in the tumor. But under most conditions, we typically see a correspondence that reflects microglial activation. So here's an example where a group induced a hemistroke in the rodent, and here's the uptake of the PET tracer over time. So it's the number of days on the x-axis since the stroke was induced. And then over here we have translocator protein co-localized with a label for microglia, and you can see that the time course of the change in translocator protein mimics the overall change that's reported in the PET scanning. Whereas over here, with the co-localization of an astroglial marker, we have a very different time course. So effectively, the markers that we're getting with the scanning tend to correspond to microglial activation rather than astroglial activation, at least in most of the validation paradigms that are done. So we have a tracer at KMH that several other groups are now using that is called 18-fluorinated FIBA. And there's a lot of desirable characteristics for it as a tracer. So there's a lot of work that goes into developing these technologies. So it has a high affinity for the translocator protein. It's completely selective, so it can be displaced with translocator protein blocking medications. It has metabolites, but those metabolites that are radioactive are not brain penetrant. And its kinetics are reversible in humans, meaning that when you take a scan and you look in the brain, you get an uptake initially and then you get this decline. And you need the decline in order to use the compartmental modeling to get out the results for measuring the binding, which are key. Now, there's also translocator protein in endothelial cells, but we do know that 4% of the signal that we get with PET is blood volume. Endothelial cells surround the blood vessels, so we see this as likely to be a very small component of the signal that's obtained. And I'll be talking about the markers that we get of TSPO-VT, which is translocator total distribution volume, which is a fancy name that really reflects the binding of the tracer to translocator protein that largely reflects the level of the protein. So you can look at this as an index of translocator protein level, this TSPO-VT, which you'll see in a lot of the slides ahead. So what is translocator protein imaging used in major depressive disorder? So there's been a neuroinflammatory theory of a major depressive disorder for some time, and the usefulness of the translocator protein would be one way to assess it, given that microglia have an important role in inflammation. So one of the arguments for the neuroinflammatory theory is that in the midst of sickness, so if one has an infection, typically have low mood, trouble enjoying things, lack of interest in eating food, losing weight, sleep disturbances, trouble with moving as fast as usual, a lot of symptoms that correspond to what's also seen in major depressive episodes. Also, we know that there's a high rate of getting depression symptoms in illnesses with neuroinflammation. So maybe typically in a medical illness, there might be a twofold sort of risk of having a depression, but in disease of neuroinflammation, it tends to be more in the threefold or higher range. And so it's interpreted by some that this may reflect that the neuroinflammation itself is predisposing to getting depression under certain circumstances. As well, if one stimulates the immune system under some conditions with vaccinations or interferon therapy for some illnesses, or giving lipopolysaccharide, this fatty sugar that provokes an immune response, that depressed mood is quite common thereafter. And there are peripheral markers of inflammation that are often reported to be elevated in major depressive disorder. So here's a review that we did in 2014 looking at reasonably large studies looking at common peripheral markers in the midst of major depressive episodes as compared to health, with most studies tending to report positive findings of elevations in the markers in depression versus health. Some studies finding no difference and no studies being negative at that point. And peripheral inflammation, however, does not reflect necessarily the brain. They can be interrelated, and it can be that a strong peripheral inflammation stimulus will result in inflammation in the brain, but they're not equivalent. So it's useful information, but not definitive. And then as well, while we've talked about a number of reasons why we would expect the neuroinflammation and depression, it's important to assess that, of course, in the brain itself. In the last couple of years, in 2000, or I guess last year, in 2018, there's been some more postmortem studies to look at some markers of inflammation in depression, looking at messenger RNA transcripts associated with cytokines, inflammatory markers that have been elevated in depression. But in 2015, when we started imaging, the data was more scarce. Their priority had largely been on suicide victims rather than depression, as the samples of depressed subjects tended not to be as large, although there was some positive finding in some. So we wanted to do a definitive study looking at 20 people in the midst of depression and 20 health to have larger numbers to look at this question and to look at whether translocator protein binding is elevated. So here we've got the regions on the x-axis, the level of translocator protein on the y-axis. These subjects that were compared were people who were in the midst of depressive episodes, did not have other active psychiatric illnesses at the time, were medication-free, and they were age-matched and of similar sex to those of the healthy controls. And then the reason for that kind of level of recruitment was that we really wanted to look at the effect of clinical depression on our comparison rather than other associated conditions that could happen at the same time. Another complication with this kind of scanning is that there's a single nucleotide polymorphism, so a change in the gene that affects the binding of the radiotracer to the translocated protein. So where you see the triangle or the circle reflects whether one happens to have the genotype that influences the binding of the tracer. We look at this as a nuisance variable and covariate out, so we can look at the effects of difference between people in the midst of depression and people in the midst of health. What you can see is that it occurs throughout the brain regions that we've assayed here. So all these gray matter brain regions, we see these differences. When one looks more broadly at subregions of the prefrontal cortex, again, we still get effects of differences between people in the midst of depression and healthy controls. And when it starts looking at other areas of the cortex, again, we still get differences. And in subcortical regions, again, we have differences. So the effect of the differences between groups was throughout the gray matter of the brain. We subsequently extended our sample, so we invited another 30 subjects to participate in our study that had major depressive episodes. And these people had different clinical histories from the first group. So they had histories of antidepressant use, current antidepressant use. Many had concurrent other illnesses like obsessive compulsive disorder. And we also increased our healthy sample. And we still found differences between people in the midst of depression and people in the midst of health. So this change that we found in elevated translocator protein was not only found in people who were medication-free, but is also found in people who have been treatment-resistant and not done well with previous interventions. When one looks at different regions in the brain, so here's the different prefrontal regions, we still get differences between people in the midst of depression, people in the midst of health. Similarly, in other cortex regions of the brain, we see the same sort of level of differences. And we also get differences in subcortical regions. Now, sometimes people say in psychiatry it's challenging to get replication, but in this case from that 2015 study, there's now, including us, 4 independent groups have published 30% elevations roughly in translocator protein binding, and 2 of us have replicated ourselves, us and one other group. There was a tiny initial negative study of about 10 subjects, but only half of them were actually in a depressive episode. And when they reevaluated the sample, the half that were in a depressive episode also had elevated translocator protein binding. So it's been a very consistent finding across studies and across sites. And I think that makes it really compelling when you have that level of consistency, that there's a target here that could be useful to examine in terms of how we might use this to help improve clinical care for people. So what I posted down here is a couple of groups of people with depression and a group with healthy controls in the translocator protein binding values. And this came from a different group. This came from Lee and colleagues. And their graphs are not so dissimilar from the graphs that I was showing you before, reflecting the consistency of findings across groups. Another issue is that we're interested in understanding what clinical symptoms relate to the levels of elevated translocator protein binding in the midst of depressive illness. And there's a couple of ways to conceptualize this. We do know that in other illnesses from neuropsychiatry, like Alzheimer's disease, multisystem atrophy, or Parkinson's disease, that there's greater levels of microglial activation with greater duration of illness. And there are models that have been considered for this. So when microglia are activated, sometimes they can make substances that might be harmful for neurons and cause some injury, and then the injury in the neurons then results in damage-associated molecular patterns that then stimulate microglia to then more microglia to then become activated. And microglia, when they're activated, can produce cytokines that can then cause more microglia to be activated or themselves to be activated. So there are these processes that are known to occur and are implicated in progressively greater levels of microglial activation with duration of illness. Now, unlike other neuropsychiatric illnesses, we use antidepressants a lot in treating major depressive episodes. And when one looks at the effect of selective serotonin uptake inhibitors, so serotonin-raising medications, where we've got cells in a dish, and you give the antidepressant, and then you might add a stimulating paradigm such as this lipopolysaccharide or another stimulating paradigm, that the antidepressants tend to suppress the level of microglial activation that occurs. So when we're looking at predictiveness of microglial activation in people who have major depressive episodes, we consider two factors: the duration of illness, using the model that's been seen in other illnesses, but also considering the antidepressant-free period, since antidepressants halt, at least in the dish and in microglia sitting with antidepressant and stimulating paradigms. And so what I show here in this plot is the years of untreated major depressive disorder, so from the time at which the first episode started to the time in which antidepressant treatment started, and then we also count and add on any gaps of 6 months or more where people haven't been taking antidepressants to get a measure of the duration of untreated major depressive disorder. And then here we've got the level of translocated protein at the level of the inflammation. And so what you can see is that in people, and these would be people who are experiencing major depressive episodes that I mentioned in the earlier sample of 50. So this would include people who are in a current episode, so they would be people who may not have had as good outcomes, and with longer duration of illness, they have a greater level of the inflammation. And we also see that in the anterior cingulate cortex and in the insula. Now, when we look at other brain regions, we see that throughout the brain that the longer the duration of untreated illness, the greater the level of inflammation. So here's the p-values for the prefrontal regions, the other cortex regions, subcortical regions. So the longer that the illness has been untreated and been present, the greater the level of translocated protein elevations. You can also model this looking at the level of translocated protein and then considering the total duration of the illness, but then putting a second factor into the duration of antidepressant treatment. And so the antidepressant treatment tends to work in the opposite direction at a similar sort of level of magnitude to the effect of the duration of illness. And so this is a cross-sectional study, but it's showing that within the sample that we've taken, that the longer the illness, the more the level of inflammation, but it's countered also by the antidepressant treatment. But it's not that the antidepressant treatment brings it back down to being nothing. It halts it from progressing using this cross-sectional point of view. So here what I show is that the same effect is found throughout the brain regions that we assess. So in all the prefrontal regions, the cortex regions, the subcortical regions, there's an effect of the duration of illness, but there's a counter effect of the antidepressant that matches up. So effectively, the longer people are having their depression before they start antidepressant treatment, the longer the period of time that there is, or the greater the level of translocated protein binding. Once antidepressant treatment starts, then we don't see this change occurring anymore. So here what I've shown is another plot that's got the regions on the x-axis, the translocated protein level, the level of inflammation on the y-axis, and then we've got three groups. We've got one group of healthy, one group who's had shorter durations of untreated illness, or 10 years or less, and one group that's had longer durations of untreated illness at 10 years or more. And so you can see that people who have these longer histories of untreated illness have greater translocated protein binding values as compared to the healthy controls, reflecting the greater, the index of inflammation. And again, here I've got this HAB and MAB, and that's referring to that genotype. So the circle means that one's got one kind of genotype, the triangle representing the other genotype, and these affect the radio tracer binding to the target, but we covary out that effect to really look at the effect of the different groups. So again, this also shows the same result, but this is in different regions in the cortex, other cortex regions, frontal regions, other cortex regions, and then subcortical regions. And here you can see that in the people who have depression with longer histories of not being treated, that their values tend to be higher than healthy controls or higher than people with shorter durations of treatment. So what about an obsessive compulsive disorder? So there's interesting reasons to look at inflammation obsessive compulsive disorder. One of them being that there are these case series that have been reported of children that have had group A beta hemolytic streptococci exposures, and they can get a syndrome where they get these involuntary movements temporarily, and they call Sydenham's chorea. And the thinking behind Sydenham's chorea is that there are material in the bacterial cell wall that overlaps with material in basal ganglia neurons, leading to an autoimmune reaction. And for these cases, there's been work done to reduce the level of inflammatory agents in the blood by trying to pull it out with plasmapheresis and other methods of suppressing inflammation and getting reductions in symptoms. So it's certainly a model that advocates for an autoimmune type of process in these cases of obsessive compulsive disorder. Now, on the other hand, it's not that common if you have it as serious and important, but it's not a high proportion of cases with obsessive compulsive disorder. However, we also know that in neuroinflammatory illnesses and in illnesses of autoimmune disease, that there are higher rates of anxiety and obsessive compulsive disorder. So it's also suggestive that heightened inflammatory states may predispose to obsessive compulsive disorder. So we examined in a study 20 volunteers who had obsessive compulsive disorder. They're medication-free, had no other active psychiatric illnesses at the time. We had 20 healthy controls who were of similar age. They had the similar genotypes that I mentioned before for influencing the binding of the tracer, similar age, fairly similar in terms of sex distribution and similar in terms of body mass. The people with obsessive compulsive disorder had sort of typically moderate to severe symptoms, and they were typically of reasonably early onset, with most before age 12 and the vast majority before age 18. In obsessive compulsive disorder, you get obsessions, these thoughts or impulses or images that are hard to shut out and suppress, that don't feel like they belong, and they're distressing, and they can cause anxiety. And then there are the compulsions, the repeated behaviors that people will do to try to make that anxiety go away. And this is a distribution of what we've found in our sample. It adds up to more than 100 because people typically have different kinds of obsessions going on concurrently and different compulsions going on concurrently. But it's a fairly representative sample, except that it's medication-free. So in obsessive compulsive disorder, there's a cortical striatal thalamic circuit that's implicated in causing this illness, and a confluence of imaging findings that have been reported previously, and in terms of also considering the mechanisms of how these structures work. And so that's the focus of the imaging study in terms of the regions that were selected. So here are the regions along the x-axis, and on the y-axis, the level of the translocator protein, our marker of inflammation. And you can see across the regions selected that there's about a 30% elevation in the translocator protein binding in obsessive compulsive disorder as compared to health. So here are the regions along the left, and here are the percent differences. And what's interesting is that you do see more of a difference in the cortical striatal thalamic circuit that's been implicated longstandingly in obsessive compulsive disorder, but we do also see differences throughout the rest of the brain as well. What we found is that people who had distress associated with trying to resist the compulsions tended to have the highest levels of the translocator protein binding in the orbital frontal cortex, that frontal area of the brain that was in red in the slide a couple before. So how can we use this information to improve treatment and help bring about more cure? So the first question is, does it matter if you get a 30% magnitude of difference? Well, we know that microglial activation occurs in Alzheimer's disease, and we know that elevated translocator protein binding occurs in Alzheimer's disease, and we even know with our specific technique that we get magnitudes that are around the 30% range in terms of differences between Alzheimer's disease and health. So yeah, so 30% magnitude is meaningful because it's consistent with the positive control of the Alzheimer's disease data. Now, another question that might be raised is, well, we don't have regional specificity in major depressive episodes. We have this finding of elevated translocator protein throughout the gray matter of the brain. And that's not necessarily surprising because there are a number of paradigms that can induce greater microglial activation throughout the brain. And here's an example of giving lipopolysaccharide to people that another group had done. And what they found was that temporarily the translocator protein bindings went up, and it was pretty similar throughout most regions of the brain. So there's certainly situations that can occur that can lead to inducing microglial activation or elevations of translocator protein that can occur throughout the brain. Now, even though these changes occur throughout the brain, there are circuits that are implicated in acting differently in the midst of major depressive episodes, as well as in anxiety disorders, of course, that we've talked about. And these regions are inclusive within the gray matter regions that have inflammation. So even though the inflammation might extend beyond the regions and circuits that are implicated in major depressive disorder, it's certainly quite feasible to conceptually that as long as the regions that are implicated in the circuits have inflammation, that they could potentially be influenced by this inflammation and lead to illness. Now, in obsessive compulsive disorder, I pointed out that the circuits were more favorably affected by unfortunately with the translocator protein, but we also have the findings throughout the other rest of the brain. And it may be that because one gets inflammation in one area of the brain, that it might also occur in other areas of the brain. So if this is a response to injury in obsessive compulsive disorder, and the microglia are there, they can produce some products that might contribute somewhat to the injury, and those products can spread around to other microglia in other areas of the brain. So it's not uncommon to find that there's inflammation in some areas of the brain and it spreads to other areas of the brain. And microglia can also, as I mentioned before, make cytokines that then can spread to other areas of the brain. So the distribution tends to be preferential to the corticostriatal thalamic circuit, but the way that the microglia function, it's a little bit like a system that, if one area of your brain needs microglial activation, the rest of your brain can certainly get prepared to deal with helping address the issues that might require microglial activation as well. These studies don't address the cause of why we have the microglial activation, and there's different reasons that this could be occurring. So certainly microglia are activated in response to trauma infection, stress that can happen in earlier phases in life, can provoke microglial activation. Exposures to infections earlier in life can prime microglia to make them more responsive to be activated. And as I talked about before, there are persistence mechanisms such as microglia producing some things that can predispose to some level of injury, and then that injury can then lead to activating more microglia. And as well, we can have the issue that neurons could be injured in the course of an illness, and then as a result of that, we could have more microglial activation. I think the latter issue is particularly important because it offers some opportunity for intervention. So sometimes in major depressive episodes, people do really well. They recover and they go on with their lives, and it's a one episode situation, and things work out fine. Unfortunately, some people will have different outcomes where they'll have recurrence of illness, and these recurrences might get more severe or become more persistent. And to date, we've had difficulty identifying biological changes in the midst of clinical depressions that increase with magnitude with increasing illness. And in this case, we've found a strong relationship between this particular marker and greater duration of illness. So it certainly could be that this is an attractive marker to target to try to see if we can come up with new cures to help people recover. This also argues to the point that short durations of clinical depression may not necessarily be the same illness at the biological level than long durations of clinical depression. And so we have to think about how do we treat short durations of clinical depression and long durations of clinical depression, and that we have to consider developing therapeutics that differentiate this. Now, what about microglial activation? What would one want to do about having microglial activation? And there are pros and cons to microglial activation. So microglia can sometimes do things that are unhelpful. When they're activated, they can make hydrogen peroxide. They can make cytokines that augment inflammation. They can make prostaglandins that are thought to sometimes contribute to lower mood. Also, in the midst of microglial activation, they can produce cytokines that then provoke a different regulation of tryptophan away from becoming serotonin towards a pathway of becoming chirenin and going down this route through inducing upregulation of 2,3-indolamine dioxygenase. Also, microglia can sometimes result in excessive synaptic pruning, so there can be downsides to microglial activation. On the other hand, there can also be protective features of microglial activation. So microglia can sometimes produce cytokines that then provoke microglia to take on curative roles, such as pruning unwanted synapses, phagocytosing and removing debris, promoting vascularization, or meaning more blood vessel promotion, increasing synaptogenesis. So there are pros and cons to having microglial activation. I think ultimately what we want to do is try to have strategies that promote more protective results and reduce the level of unhelpful results from having microglial activation. The translocator protein imaging that we're receiving, it gives us a measure that there's a morphological change. So the microglia have changed their shape. It doesn't tell us to what extent they work down one direction or another direction. However, we know that when we induce microglial activation with lipopolysaccharide or vaccinations, that this is associated with depression symptoms. It also can be associated with anxiety symptoms. So there's lots of reason to think that we probably want to interfere with some components of microglial activation in order to try to make novel cures. And we know that, as I mentioned before, that illnesses that are associated with greater microglial activation also have higher rates of comorbid depression and anxiety and obsessive compulsive disorder. So there's reasons to think that one would want to target this. And companies have felt the same way. So they see these imaging studies and they recognize that there's a third of people who have clinical depression who might potentially have a target that they could then look to therapeutically intervene. So a couple of directions that have been looked at. One is there is development of inhibitors of proliferation of microglia. There's also strategies to interfere with that 2,3-indolamine dioxygenase so as to divert tryptophan away from the chirenin pathway and to move down towards becoming serotonin instead. And then there's academics. This is something that we're also involved in, which is to repurpose medications that are on the market for other indications and bring them towards either reducing microglial activation or shifting microglia to curative roles. So minocycline has been implicated in both of these activities. And so that's an interesting target or intervention that we're evaluating. And another one that's been looked at is to look at COX-2 inhibitors, such as celecoxib, which is on the market to treat arthritis. Now, I'm not suggesting that one should go out and take minocycline and a COX-2 inhibitor right away or anything like that. I think it's important that these things are studied carefully in clinical trials, but it does raise the possibility that there could be interventions that are around the corner if we match these up to the right types of illness. So one of the challenges is that we have key brain systems that are affected by illness that I've talked about a bit before. And then we think that there's multiple ways that there can be altered neural injuries or neurochemical changes that are affecting these systems. And when one's looking at trying to make new medications to target something like microglial activation, a key issue is how can you identify the subtypes of illness that are the best match. And so one thing we've tried to do is identify symptoms, and I mentioned a couple of symptoms such as, or clinical phenomena in the case of depression, that longer duration of untreated illness is one predictor. In the case of obsessive compulsive disorder, the resistance to, or the difficulty resisting the obsessions and difficulty resisting compulsions in particular are an area that's been a phenomenon that's been associated with greater translocated protein. But those symptom correlations don't look quite predictive enough for optimally what we would like. So we're also looking at blood predictors. And so there's been different efforts to look at blood markers of inflammation. I reviewed a few of them earlier. And one of the thoughts is that because inflammation in the periphery can affect the brain, that blood markers that it could be directly related to markers of inflammation in the brain. So a number of mechanisms have been implicated. Direct effects of molecular patterns reflecting inflammation that can stimulate the vagal nerve directly. There are also cytokines can be transported across the blood-brain barrier. There are cells that function in an immune response way near the ventricles, which is the idea behind this picture here. And they can release cytokines into the brain, are influenced by the local environment as well. However, when we've looked at these more straightforward markers of blood inflammation, they haven't related well to the translocator protein binding. So here's a translocator protein binding in a few different regions and various markers, and we didn't get the positive correlations that we had hoped for on this initial go. And that could certainly be that the reason is that at the time that we're doing the scan, it might be not be the time when there's profound inflammation in the body. It could be that inflammation in the brain is related to repeated episodes of inflammation in the body, and that what one gets at the time of the scan isn't necessarily the best match. And here's an example making the point that inflammation marker in the blood at age 9 was predictive of a clinical depression at age 18. So it shows that sometimes in an empirical way that inflammation at an earlier stage in life might be related to getting depression later in life. And we talked earlier about markers and methods by which microglial activation can perpetuate itself over longer periods of time through autocrine effects as well as through influencing nearby neurons. So what we're looking at in terms of developing blood markers is to look at products that are made by microglia that go out into the blood. And then what we are then looking at is how we can correct for this by considering peripheral contributions to the same marker. And so we have some composite markers that look really promising. I can't show them because they're not published yet, and I can only show you the published data, but I can say I think there's promise in this direction, and we're still moving ahead with this. So I've talked about PET translocated protein imaging, and we use the translocated protein imaging to try to help stratify who with major depressive episodes and obsessive compulsive disorder has this particular phenomena that we think reflects microglial activation primarily. We're trying to develop low-cost predictors, and that's in process. We are assessing, and others are assessing the impact of therapeutics. I give the example of repurposed medications, but there's also medications being developed by industry. And what the idea is that in the longer run, what we'll be doing, and we actually are in the process of doing this with some repurposed medications, is seeing whether greater translocator protein binding in the midst of depressive episodes will predict response to medications that influence brain inflammation or microglial activation. Now, because microglial activation is a complicated phenomenon, it's typical in postmortem studies to try to identify multiple markers, and to, as well, in a consistent way, in PET imaging, they're looking at other markers that can be indicative of microglial activation, and as well, there are a number of markers being looked at for other processes of inflammation, such as astroglial activation and so on. And astrocytes are about 20% of brain cells. They activate in response to infection or trauma and in neurodegenerative disease. However, this is an example that demonstrates some of the challenges with this. So monoamine oxidase B is a target that's overexpressed in astrocytes in illness. And so it might be an attractive marker of astroglial activation, which I think it is, but it's also expressed in serotonin-releasing neurons. Stress hormones can induce greater levels of monoamine oxidase E and induce transcription of monoamine oxidase B. So it's not solely a marker of inflammatory response of astroglial activation, but it is an interesting target because it metabolizes dopamine, norepinephrine. It can contribute to creating hydrogen peroxide, and it participates in programmed cell death. So there's a lot of things that would be appealing about this target in mood disorders and potentially anxiety disorders as well. So I give this as an example. It's a tracer that's come a long ways in development, and it has a lot of attractive qualities. High affinity for MAO-B, it's selective, doesn't have brain-penetrant metabolites. It's got this reversibility, meaning we get the scan time, we get uptake of the tracer and the decline over time. So it's got a lot of the qualities that we really like in a tracer, and there are a number of these sort of technologies that are in development. And I think that this reflects that there's really an exciting time for developing new technologies. And as we get a greater understanding of the nuances of the inflammation changes that are occurring, we can come up with better and more targeted treatments. So my main points are that the translocator protein PET imaging mainly reflects microglial activation, an important component of neuroinflammation, that there's greater translocator protein binding throughout the gray matter in clinical depression, but it includes mood-influencing brain circuits, and it's been well replicated. The TSPO distribution volume values, the binding, is highest in clinical depression with longer durations of untreated illness, and it represents an example of neuroprogression in cases of clinical depression that have not gone as well. It doesn't necessarily reflect cases of clinical depression that have gone well, but rather in ones that have not. Greater translocator protein binding also occurs in obsessive compulsive disorder, and it's found, but it's also found throughout the gray matter. We do see it more prominently in the implicated corticostriatal thalamic circuit. The imaging findings make a case for developing new therapeutics to target microglial activation or their adverse sequelae, and to try to shift microglia into becoming more beneficial helpers in the brain. And key future directions are that there's ongoing work to develop blood markers that can predict elevated levels of translocator protein binding in the brain. Again, also work to look at whether translocator protein binding can be predictive of response to inflammatory modulating interventions. And there's an ongoing development of a number of new markers. So you saw there's quite an exhaustive list there that can be used to better characterize microglial activation and as well to characterize other aspects of neuroinflammation. So perhaps I could just acknowledge a number of collaborators and take a couple of questions.
Well, Jeff, thank you for an excellent presentation. A lot of information there, very interesting and very important and promising. I guess one of the questions that a few people have alluded to is right here and now, and to nowadays, what can somebody who hasn't responded to the traditional treatments do with regards to any of the information that you just provided? Are there trials, are there approaches that people can take related to the issue of inflammation?
Oh, that's an excellent question. If one looks at clinicaltrials.gov, that would be the place to see what kind of trials are available. Some of the trials that are going on are to look at adding minocycline on to conventional antidepressants. As well, there are also trials looking at celecoxib. So those are trials that happen to be going on in my region, but I'm sure that there are other ones going on in other regions. And so those would be examples. As well, there are companies developing inflammatory modulating treatments. So again, the clinicaltrials.gov would be the way to localize what would be available in the region that inquirers are living.
Okay. Have you looked at other aspects such as diet or exercise and how that may affect inflammation in people?
Well, I think that's certainly an important question. Right now, what we No, we don't. The best way to look at this sort of question would be ideally to look at effects of exercise and diet on the translocated protein. And while we haven't done that, what we do know in general is that peripheral inflammation over time has effects on the brain, at least in a lot of different models. So I think in the absence of having imaging that directly measures the effects of exercise or certain lifestyle changes, that maybe the best thing to do is to consider what strategies can be used to lower your general bodily inflammation. And then one strategy is that having a healthy, more Mediterranean type diet with more fruits and vegetables and regular exercise would be a way to lower body fat, and then lowering body fat generally lowers bodily inflammation. But I wouldn't be able to make the claim that that's been tested yet, that lowering bodily inflammation equals lowering brain inflammation, is that may take time. But another strategy that might also be considered is that inflammation in the brain is implicated in causing pro-oxidant effects. So it could be that in diets having antioxidant effects could be useful. But I think for all these interventions, there are probably things that one would want to do in the here and now that are just generally healthy anyways. But in the longer run, what we hope is that we'll be able to look at effects of exercise, diet more carefully on these brain imaging measures and intervention studies.
So at this point, there isn't evidence to say follow these kind of what's viewed as healthy diets or exercise regimens. There's no evidence that would have an effect, but it's good to do that anyway, and perhaps down the road there will be evidence that it has an effect. So certainly it makes sense to take those steps, both as a preventative and as an adjunct to usual treatments.
I think that's fair to say. I think that right now, in the way that the science has developed, is that people have chosen what we think are the strongest interventions to assess. So a lot of the work has been to look at the minocycline intervention, because people think that's probably a stronger intervention to affect brain inflammation. Then people move into other strategies to see whether they may also affect brain inflammation. And just because there isn't evidence doesn't mean that there's negative evidence. It just hasn't been studied enough yet because the technologies are still quite new.
How does this affect the issue of prevention? Do you see being able to develop these biomarkers that may say that someone's at higher risk, and therefore certain interventions could be made to decrease the risk of developing depression or OCD?
I think that's certainly possible. I think that right now, I'm most optimistic that when someone has a first episode of depression, that there may be opportunity at that point to try to develop strategies to instruct the microglia to be more curative at the beginning of a depressive episode. In terms of avoiding provoking microglial activation itself, that could also have potential. One could look at things like nutritional supplements and try to look at inflammatory provoking stimuli and seeing whether one can prevent them with certain dietary interventions. So that could be potentially an area that could develop in time. So it's something that I think there's potential for. It would take some carefully designed studies, but it certainly could be done.
And what do you see as a time frame? And I know you can't give any guarantees, but what do you see happening over the next 3, 5, 10 years in this area?
I think that it's likely that there will be therapeutics for people who have more highly treatment-resistant depressions that target inflammation. I think it's likely we'll develop predictors of who is going to have that inflammation, and that's probably where the first wave of effort will go. I think in longer time periods, then there will be assessments on lifestyle and other issues in terms of how they can affect the inflammation in the brain as well. But I think that probably the first wave of work will be advancing treatments that target the inflammation, because it's more prevalent in people who have longer histories of depression and in somewhat more severe OCD, at least with regards to having difficulty resisting those compulsions. So I think that where the inflammation is showing up the most prominently is probably where the most therapeutics will start, and then I think from there it will go into earlier and earlier stages in terms of aspects of earlier points in illness, and then before illness and prevention.
Our time is just about up, so I want to say thank you again for taking the time to present to us, and more importantly, thank you for your ongoing work in this important area that I think will have a tremendous impact. I also want to thank everybody who joined us today. All of the research we fund is made possible through private donations, so please consider making a contribution by visiting our website, bbrfoundation.org, or calling us at 800-829-8289. This webinar has been recorded. If you've missed any portion or would like to share it with a family member or friend, please visit the events and webinar page on our website. I hope you'll join us again in January 2019 when Dr. John Mann, the Paul Jansen Professor of Translational Neuroscience and Psychiatry and Radiology at Columbia University Medical Center and Director of Research and Molecular Imaging at the New York State Psychiatric Institute, will present Brain Plasticity, the Effects of Antidepressants on Major Depression. This webinar will take place on Tuesday, January 8th at 2 p.m. Eastern Time. Thank you again for joining us, and I wish you a happy holiday season and a happy and a healthy new year. Take care.